Okay. Okay, so yeah, Aki, maybe you can share your screen now.
Yes.
Okay, good. Okay, so good afternoon, everyone, and good morning for Akim. It is my pleasure to introduce our esteemed speaker today. Actually, it's the last speaker of this colloquium series this semester, Professor Achim Kramer. So, Dr. Kramer is a leading chronobiologist and a renowned expert in the field of circadian biology, with a distinguished career at Charité University in Berlin, where he served as a professor and leads groundbreaking research on the molecular mechanism governing biological rhythm. So I still remember that when I was a grad student, he was already very famous faculty and giving a great talk, and it was always inspiring me. In particular, Akim not only performed the experiment, he also do the theoretical work together. So his work is one of the example of truly interdisciplinary approach for the circadian rhythm. So it'll be a great opportunity for our group to see his research. So let's welcome Achim Kramer.
Thank you, Jay, for the really nice introduction. And I'm looking forward to presenting some of the work we have been done in the last years
Mhm.
to your group and your colleagues.
Mhm.
Um, I, since I was told that not everyone has a strong background in circadian biology, I do a brief introduction into circadian rhythms to get everyone on the same
Mhm.
page. And I start with just reminding everyone about this oscillator, which has a 24-hour period, at least now, and at the times when life evolved was a bit shorter, but the bottom line is that it created oscillations in the environment, including light dark cycles, temperature cycles, availability of food, and so on and so forth. And this oscillating environment triggered the evolution of internal timing systems of organisms which are exposed to that oscillating environment in order to let them anticipate these recurring chances and challenges in the environment, to be able to be prepared for that and to be fitter than others. So it's an external oscillator which drove the evolution of an internal oscillator, our internal circadian clock. And what I mean with anticipation, I would like to illustrate by showing you this time-lapsed video of a sunflower, which still does not bloom yet, but is still growing. And as you know, plants get their energy by doing photosynthesis. And to do that most efficiently, they have to at least the sunflower, they orient their leaves towards the sun to catch as many photons as possible. And there's two possibilities. So I show you this video now, and you see the sun is kind of not perfectly aligned to my video, but this is my own PowerPoint animation. But you see over the course of the day, the leaves orient from east to west to be able to capture most of the photons. But the interesting part comes now during the night. Now it's night,
Mhm. Mhm.
and then in the second half of the night, you see that the leaves are orienting already towards east in anticipation that the sun is coming up in the east. So there is this is not a so the leaf movement is not a passive reaction with respect to light exposure, but it's already anticipating the upcoming light in the next couple hours. So there is this is driven by molecular circadian clocks in the stem of the sunflower, and with opposing phases in the eastern part and the western part of the stem. I don't go into detail here. The point is that this organism uses its internal clock to anticipate the next chance, which is getting as many photons as possible, and prepares for them. And this is what we also do in our body. For example, we have a high amplitude hormone rhythm with respect to cortisol, which peaks at the beginning of the day in humans
Oh,
and has it.
okay. It looks like the slide didn't move. Can you? We are
Well,
still.
I'm. This is not.
Oh,
I'm.
yeah, yeah. I see.
I don't have a
Sorry.
slide
Yes.
for this.
Okay, gotcha. So I thought that there is a
So
problem with your slide.
I have
Okay.
no slide for the cortisol, but I just wanted
Yeah.
to mention that cortisol
Mm,
has its trough in the evening, and in the
mm,
second half of the night, similar to this leaf movement, it rises to prepare humans to get from a horizontal into a vertical position in the morning, which requires high blood pressure and high heart rate, which cortisol promotes. So just a few terms and hallmarks about circadian rhythms. In humans, of course, we all know the sleep-wake cycle is one of the key outputs of our behaviors, which have a 24-hour rhythm. In animals, in laboratory animals, you can
mhm,
easily measure activity as one of the behavioral outputs of the clock, and you can do that by fixing a magnetic switch on the running wheel of in a mouse or hamster in this case, and then you can, similar to the to the variable data you get from your humans, you can plot that in a so-called actogram where each line is one 24-hour cycle. In this case, we have 12 hours light, the white bar, and 12 hours darkness in the animal facility. And you can easily see that this hamster is um, has most of its activity during the dark phase, to the second half of the night. It's a nocturnal animal, and you can also see it's perfectly aligned to the light-dark cycle. So always, if the light is switched off in the animal facility, the animal starts running for several kilometers per night because they love the running wheel. But the key point is now what happens if we switch off the light completely? So, because that could be interpreted as a passive reaction to the light. So,
mhm,
meaning that maybe one hypothesis would be the hamster just doesn't like the light and does not move during the light phase.
mhm,
And when the light is switched off, it will move.
mhm,
However, and to test that, whether this is just a driven behavior or an endogenous behavior, we remove all time information from this animal, which means just switching off the light. And this is what happens. What you see here in constant darkness, no time information. So everything is the same, constant temperature, constant air pressure, constant noise levels, constant darkness. You can still see rhythmicity persists. So we have phases of hours of inactivity followed by hours of activity. But what is striking here that the animal every day um, jumps into the running wheel half an hour earlier, which means that the period length of this oscillation is shorter than 24 hours. In this case, maybe 23.5 hours, which also implies this is not driven by a mysterious factor X, which we are maybe unable to shield the animal from, for example, maybe magnetic field or so, because this is definitely not a 24-hour oscillation, implying that it's not mysteriously kind of exposed by Mother Earth to this animal. But this is clearly an internal endogenous rhythm. And the period is not exactly 24 hours, and hence the term circadian can be coined to this rhythm because circadian means about for circa, and dies is Latin and means a day. So the period length is not exactly one day, but only approximately about one day, hence the term circadian. So we have an endogenous rhythm, which is not exactly 24 hours long, however, can be, and we call it entrainment, can be entrained by light-dark cycles to exactly 24 hours. And a fourth property is that it is a self-sustained rhythm, which means this rhythmicity persists as long as the animal lived. So it's not a damped oscillator, it's a self-sustained oscillator, which is driven by a molecular mechanism, with what we see in a minute. Just to let you know that there were famous experiments with humans asking the questions whether also humans have a circadian clock, mostly done by a German chronobiologist, Jürgen Aschoff, using first, Second World War bunkers in Munich, and then specially built time isolation apartments, which were shielded from light, from temperature fluctuations, from noise levels, and so in the 60s, in the last century, kind of volunteers spent weeks in these time isolation apartments, maybe writing their master thesis or whatever. Complete time isolation, however, not complete darkness, as you can imagine, because humans don't want to live for weeks in constant darkness, because then they cannot write their thesis or work. So they could self-select their light-dark cycles, and then they have rectal probes for temperature, and they kind of push buttons to tell when they go to sleep and so on. And next door, researchers kind of took all this data, and these are the time series data modelers and data scientists of that days were been analyzing and looking for circadian rhythms, and they found circadian rhythms even in time isolation, which were in humans mostly longer than 24 hours, sometimes even completely really long 40-hour days. So people were complaining when they came out of this apartment because they have been made a deal, you stay 4 weeks, and then apparently researchers opened the apartment after 3 weeks, and then they said, oh, it's only 3 weeks. I have to write my discussion chapter, for example, for my
So Achim, so in this
and
experiments, it was not complete darkness. They can freely turn on the light or off whenever they want.
exactly. And
Ah, then
that was later interpreted as one of the reasons why the apparent period was so long. So
so long, right?
for a
I see.
while, people thought, oh, it's
Mm,
25 hours for humans, but then it's clear because humans often delay their
mm,
bedtimes
mm,
until the very last moment when the body temperature is minimum, and then they kind of expose their phase delay part
yeah,
more than the phase advance part. So
I see.
the period was longer than when you
Ah,
do that in a protocol which is called forced desynchrony, which
I
Chuck
see.
Seiss did. Then the average period, endogenous period for humans was 24.12 hours.
Yes,
So
yes,
we are still have longer than 24-hour period, but not as long as was suggested in these experiments.
I see.
The
Because
bottom line is
whenever I see it, it's good. Thank you.
that humans also have an internal clock, and the question is now, how is that driven? And maybe before I show you that, the it is now clear that we have circadian rhythms have a genetic basis, because we know from humans and animals with mutations and all the clock genes, and which determine the circadian period. And we also have a good feeling about the anatomical um, location of the master clock, which is in the suprachiasmatic nucleus in the brain, which is below the eyes, in the middle of the brain, maybe a couple centimeter um, below this point. And if you destroy this so-called suprachiasmatic nucleus in the brain, you don't have these rhythms anymore. The molecular mechanism is grossly simplified here, and I'll show you a more accurate picture in a minute. The point is, for youth, people who are good in theory, they know that negative feedback loops, which have a delay, can lead to oscillations, bifurcations, oscillations. And this is exactly how the molecular mechanism of the circadian clock in principle works. So we have negative elements, clock proteins, clock genes, which are called period and cryptochrome genes. There are more, but this is just for the sake of simplicity here. And we will even more simplify it by showing only one per and one cry in a second when I switch off this on this movie. Then we have transcription factors, which are called BMAL1 and clock, which activate the transcription of these genes. And these genes are then transcribed into mRNA and translated into proteins. And the negative feedback, now I switch on, so only one per. The negative feedback is these proteins at some point in time after several hours. So they do a lot of post-translational modifications and complex formations. And this is probably all wrong, what I show here, but the principle is right, that these cry and per proteins at some point in time inhibit their own positive activators, clock and BMAL, by blocking them their transcriptional activity. And then, since these proteins also have a certain half-life, several hours later, they will be all, this concentration goes down and they will be all degraded and the next cycle can start. So this has been mainly found out in Drosophila, and in the 1990s of the last century mostly, and Nobel Prize for Medicine has been awarded to these three scientists, US scientists working on the molecular mechanism in Drosophila circadian clocks, Jeff Hall, Michael Rosbach, and Michael Young. So the situation is in reality more complicated and probably resembles more these detailed picture, and it's probably even more complicated. So you have here the per and the clock and the BMS. This is the nucleus. You have the complex formation, and Jay, your group has been working on phosphorylation and the impact on the different phosphorylation sites, on the period, on the stability, and you know that there are, depending on the type of phosphorylation, there are several properties. They have been even implicated in temperature compensation, a property which I did not mention yet. And degradation and phosphorylation is key for the circadian period. And this is what we have been learning from a lot of genetics and a lot of biochemistry, often with a kind of liver tissue material and a lot of Western blots, and also in the circadian field we use a lot of these reporter assays with luciferase hooked to clock clean promoters, and what is, however, not so clear is the temporal dynamics of all these events. So we don't really know when these proteins go back to the nucleus and what property do they have, and how big the complex is. I mean, there are very good biochemistry papers based on liver tissue from, for example, from the Chuck White's lab, where it has been proposed that it's not a one per and one cry protein which complex together, but it's probably a big molecular machine with per and cry complexes having a size of 1.9 megadalton, consisting of dozens of proteins, including all the pers, all the cries. And how these complexes are formed and when is not so clear. So that's why Christian, a postdoc in my lab, and now we come to the first part of the data, combines several tricks of CRISPR-Cas9 genome editing to add fluorescence tags to reporters to the endogenous clock proteins. And we use a human cell line, which is U2S, called U2S, which is a workhorse in the circadian field. Because what we want to see is in single cells, we want to observe the dynamics and the location of the proteins and of endogenous proteins. And that's why we need to tag endogenous proteins, which is not easy with proteins which are sometimes not even detectable. These tricks were necessary because these clock proteins are expressed transiently and at low levels, and that made standard techniques inefficient and unsuccessful. And I don't want to go into detail here. The knocking strategy is published. So Christian created several reporter lines for CRY1 and CRY2 and PER1 and PER2 with different tags. Some of them are red, some of them are green. And also we use luciferase knock-ins, and you can see this nicely oscillate in abundance. And this is not overexpressed, this is endogenous proteins. So now you can using sophisticated microscope, you can analyze many single cells, and what he found is that CRY1 is the least rhythmic protein, which with a much lower percentage, depending on the p-value cutoff, which is here on the y-axis, and this is percentage rhythmicity, and as you can see, the CRYs are least rhythmic compared to the CRY1 is least rhythmic and compared to PER1 and CRY2. And also, we knew already from single-cell experiments with luciferase reporters that all dynamic parameters vary widely. So the period ranges from the single cells from 18 hours to 30 hours. And the relative amplitudes are also highly variable between cells. And consistent with the lowest rhythmicity, CRY1 has the lowest amplitudes, but the highest expression levels. So this is expression, which is you can predict that with mathematical modeling. If you have a highly, it's probably most stable, and therefore the amplitude is lowest. And now, since we can also make double knock-ins, we can quantify the phase relation between clock proteins. And this is also highly variable in single cells. On average, CRY1 is 2 hours later than CRY2, and this is the spread. And we see that CRY1 is 5 hours later than PER2 and 3 hours later than PER1. And in U2S, this is our phase map of the proteins, which has been not PER2 peaks first, then PER1, CRY2, and the latest is CRY1. And with these new tools, we can now approach fundamental questions of chronobiology in a completely new way, because we can exploit this cellular heterogeneity without the need of performing perturbations. In other words, because we have so many different cells with so many different properties, we can ask questions, for example, is period related to stability without the need of making mutations? So again, the prevailing concept in chronobiology is that the stability of the negative components of the circadian feedback loop is a key determinant of the circadian period. And that means the more stable the negative element Z is, the longer the period should be. And many mathematical models support this concept, and more importantly, a vast amount of experimental data. For example, the stability period correlation of different frequency mutants in ROSPAR. This is the negative element in this fungus circadian clock. The longer the half-life, the longer the period of these different mutants. And this also has been shown for casein kinase mutants affecting period stability in flies. Double time short period, and so on. So, however, there are also reports for example, by the laboratories of Dunlap and Ueda that find a decoupling of stability and period,
Yes.
suggesting, for example, for these mutants,
Mm-hmm.
suggesting that not the quantity, but the quality, for example, phosphorylation state, governs the circadian period.
Mm-hmm.
However, these findings were all based on studying different mutants in the negative elements. And with the ability of now monitoring single cell rhythms, we can now exploit the fact that this is a there is a broad variability in the circadian period, and probably due to heterogeneity of molecular processes in single cells. That is, we can ask the question, is period correlated with negative element stability in single wild type cells? And to do this, we did the following experiment. We used unsynchronized clock reporter cells with these knock-ins and treated after several days of free run with a cyclohexamide. And cyclohexamide blocks translation. And so we can measure both the circadian period in the first phase of the experiment and the
Mm-hmm.
degradation rate
Oh,
um, of the clock protein within hundreds of cells in parallel.
oh.
Um, so as a proof of concept to check whether this works, we knocked down FBXL3, which is a known key determinant of the stability of cry. And we saw in various reporter cells that, as expected, the period lengthens because the
Mm.
rise gets more stable. So, but only the protein half-life we can also detect, which is higher in the cries, but is largely unaffected in the per cells. So that
Right.
means the difference in stability and period can be faithfully detected at the single cell level.
Wow.
So since we did not synchronize our cells, we hit them with the translation blocker
Mm-hmm.
at all possible phases and
Wow.
find that cry1 and cry2 and for1,
Mm-hmm.
the
Mm-hmm.
stability depends on the phase.
Wow.
As it has been already described by the Fukada lab for PER2.
Mm-hmm.
The half-life of these negative elements is higher at times when the protein levels are lowest,
Wow.
um, or when they are in the falling phase. As you can see from these shaded areas, we again see a large heterogeneity of stabilities in the single cells. And this is now the opportunity. We can now test whether stability and circadian period correlate. So, and surprisingly, they do not. So, longer periods in single cells do not come with higher stability in the very same cells, here shown for cryo-reporter cells. This is
Mm-hmm.
also true for the other clock proteins tested. But this is only half of the story.
Mm-hmm.
Interestingly, when we analyze this relationship at different phases,
Mm-hmm.
we do find significant correlations.
Oh.
At the peak phase, long periods are correlated with low half-lives,
Mm-hmm.
and 4 hours later, no correlation is detected. And 10 hours after the peak, after the cry
Mm-hmm.
peak, this is the falling phase of cry abundance, there is a positive correlation between stability and period. While in the rising phase, the correlation is negative. And this is summarized in this plot, where you can see negative correlations in situations 1 and 4. This is here and here. The rising, and this is the rising phase of cry abundance and the peak phase. And a positive correlation, this is situation 3, where you have the falling phase.
Mm-hmm.
So the conceptual model is like this. Um, High stability during the rising phase accelerates the period, probably because the critical concentration is reached faster.
Mm-hmm.
Whereas high stability in the falling phase decelerates the clock during a prolonged feedback repression.
Mm.
So that's my first data summary Endogenous clock proteins reporters allow studying their subcellular subcellular dynamics. Cry is the least rhythmic, but the most abundant negative element. Phase relations, periods, and amplitudes and stabilities are highly variable, and the period is correlated with the stability of cry 1 and cry 2 and per 1, but in a phase-dependent manner. So,
Akim, sorry, maybe before it, any question for his first part? It was already very exciting to see it. Oh, so sort
Can
of,
I ask
yeah, Umin, go ahead.
can I ask question?
Oh,
I just wonder whether you distinguish the cytoplasmic protein and nucleic protein. For example,
for the half-life?
oh yeah,
yeah.
yeah.
Yeah, we're testing the nuclear protein only at this moment because it's more all the cries we see cries mostly in the nucleus and almost never in the cytosol,
Ah,
interestingly. So detectable, which doesn't mean it's not there, but we cannot faithfully detect it. Whereas for the per proteins, we often see also cytosolic proteins, but more in the nucleus, and we focus here on the nuclear stability.
thank you.
I should have said that, yes.
Is it good? What do you mean?
Yeah.
And I also
Okay.
have a one short
Sure.
question. I might
Sure, yeah.
miss, but so it's very interesting concept, the phase-specific half-life. So actually, where does this phase-specific half-life difference stem from? I mean, some specific molecular
Molecular
mechanism.
mechanism?
Yeah,
Yeah, I think we don't know exactly, but the obvious reason might be that the degree of phosphorylation, which changes over the circadian cycle,
Yeah.
will determine whether, for example, a protein is amenable for being recognized by F-box proteins.
I see.
So we know that hyperphosphorylated pairs, for example, are more prone to degradation, are now predicted to have a lower half-life.
I see. Post-translational mechanisms may be related to this specific
Yes.
phase-specific half-life. I see.
Yes.
Thank
Okay.
you.
Great. Any question? Okay. Akim, you can move forward.
Thank you for the questions. They are very good. So we have had a large number of heterogeneous oscillators, which were very useful in answering fundamental questions of chronobiology, because we had these different abilities, we have different half-lives, and we can also now study the phase relationships of the clock proteins, whether they affect the period and half-lives. But now let me turn to the heterogeneity of the human population and the insights then that can be gained from it. As you probably all know, the phase of entrainment, which is also known as our chronotype, whether we are morning larks or night owls, and fortunately I'm not a night owl because in Berlin, Germany, it's now 8 o'clock, and this is a time when I would normally start working. So I'm perfectly aligned with your working day, fortunately, but late types may still be in bed at that time.
Yes.
And we now know from many beautiful colleagues and studies that this is determined by many factors, and probably you can appreciate based on what we heard in the last half an hour that the genes play a role for the period, and of course then also for the phase of entrainment, which means the phase. So longer periods would result in a late phase, and shorter period would result into an early phase. But we also know that sex plays a role, and women are often earlier than men, and age plays a role. Younger people, teenagers, are the latest. and older people are earlier. This is based on just questionnaire data when you are, and probably also from variable data. I don't know whether they are, you probably know better than me whether there are large epidemiological variable data and analyzed with respect to sex and age, and whether you also see these influence factors. Zeitgeber strength means basically how much light you get will also influence your phase of entrainment. And of course, the phase of entrainment is often reported in social time, meaning in Korea, it's now 4:30 in the afternoon. And depending on where you are in this time zone, your phase of entrainment reported in social time may be earlier or later. So to measure the phase of entrainment, scientists and physicians use various methods ranging from questionnaires over actigraphy to the so-called dim light melatonin onset, which is considered the gold standard for phase of entrainment in humans. It measures the rise of melatonin secretion in the evening in blood or saliva. And this is complicated and requires many samples and special settings. You need to sample a lot of biospecimens over an extended period of time. It needs to be in the evening, and it needs to be in dim light conditions, because light has the property to acutely suppress melatonin secretion. So this is complicated and is only done in academic study settings in research, but not in medicine and not in the field. To more easily, but at the same time, to objectively measure the phase of entrainment, we have developed a blood-based assay, which needs only one blood sample to tell circadian phase. And since this is published, and I show you a similar assay for another biospecimen, I just want to mention it here and can answer questions later. What we do is we have been identified the biomarker genes from time series data and ask computer algorithms to extract the time telling features, which I will show you in a minute for the other example. But what we used here was we didn't use all the blood cells. We only focused on monocytes, which have a particularly high amplitude circadian clock in comparison to other blood cells like B cells and T cells, which the clocks are not so good. And we now use about 12 genes, including many of the known clock genes, to predict internal time from a single sample. And this is the result of a validation study where we used a completely different set of humans. I think 28 humans sampled in the morning and in the afternoon. And we did both this classical melatonin gold standard DILMO assay and this blood-based single time point predicted DILMO assay. And the correlation is really nice. And this is the median absolute deviation between the two assays, which is here 0.6 hours, which is in the range of the accuracy of the melatonin assay. So really good. How we did that will be much clearer after the next 10 minutes, because I show you how we do that in a different type of biospecimens. Because this is very precise, but it has a major limitation. This blood test. It is difficult to use it in field studies. So you always need a doctor or a nurse. They have to come to the lab, and you cannot do that in field studies. Therefore, we looked for a more accessible biospecimens from humans that can be collected at home without the need of a nurse or a doctor. And the Akashi lab from Japan has published over 10 years ago that in hair root cells, there is a beautiful circadian rhythm. So we asked the question whether we can identify genes in hair root cells that are first of all, robustly rhythmic among many individuals, and secondly, have a stable phase relative to this Dilmore gold standard, and thirdly, are similar in magnitude of expression. And to test this, we sampled hair roots from 13 healthy subjects in regular 3-hour intervals over 36 hours. And prepared the RNA and analyzed the expression of 96 pre-selected genes using nanostring. In parallel, we determined this Dilmore gold standard for each subject and assigned each gene expression value a timestamp relative to Dilmore and not the clock on the wall. So this is so every sample got an internal timestamp. So we found many genes significantly rhythmic when averaging over all individuals. Here is an example of this is ref or beta, or also called NR1D2. This is BMA1, also called ARNTL. And then you can see there are many genes, and they all have kind of different phases. And you can see some have higher amplitudes and some have lower amplitudes. And this is now a couple examples, and to really now, then we ask the question. So we ask different computer algorithms to extract a panel of these genes, which are most accurately predicting each single time point by a leave one out cross-validation a method. So we used these three algorithms, a lasso model with certain parameters, lambda parameters, partially square models, mostly with 3 or 4 components, and Zeitzeiger models, which is an algorithm published by Jake Yugi, where you can also select different parameters. So it's a sparse partial principal component algorithm. And so we have 3 different models, and we use the data set with the goal to identify gene sets and parameters that are best predict the individual phases when using only a single sample. And as I said, we used leave one out cross-validation, and we came up with these 3 prediction models, which we subsequently tested. And this is important. You always need to test your biomarkers with an independent study. So we this time we took only 1 hair sample per individual, and we used, I think here, 21 individuals for the validation study. It's not a lot, but it's but we also need to have the standard Dilmore, because we need to know the real phase, and we need to know the predicted Dilmore. And these are the different models, and we can also combine them and say, let's take the circular mean of all these 3 predictions, and then you get a pretty good prediction accuracy. So again, with a medium of slightly under 1 hour accuracy. So we founded a spin-off company, and people can now buy this test. And I gave you my conflicts of interest in the first slide, body clock. So people in Germany, and I think in the German-speaking world, have been about 4,000 people have been buying this test in the meantime, and we get the data and can now kind of come up with a epidemiology of chronotype, which is not related to questionnaires. And this is the time we get is the predicted dim light melatonin onsets. And to put that into context for you is that when this Dilmore time predicts about so this is about 2 to 3 hours before your bedtime. So if you have a mean of 8:30 p.m. the mean bedtime is about 11 o'clock
Yes.
in the evening. This can also, of course, this phase relation between Dilmo and bedtime also is variable, but this is the mean. And you can also see again that there are some people who have their Dilmo time at 2 in the morning, which would predict their bedtime would be at 4 o'clock in the morning, and others are 8 hours earlier, like at bedtime at 8 o'clock in the evening, 7 o'clock in the evening, but rarely. So again, a normal distribution, which is very similar to what other people have been reporting using questionnaire data. What we also see is a difference in males and females, with males on average 15 minutes later than females, which is a little less of a difference maybe than what we know from questionnaire data, but again, this is consistent. So the age, I should maybe mention, the age range was from 18 to 89 years old. Yes, okay.
So Achim, in this histogram, one person has a one Dilmo in this histogram, or one person can have many Dilmo measurement.
No, one person has one Dilmo,
So
one biomarker-based Dilmo, yes.
I see. Okay. So we can think that it's a intrinsic chronotype.
Yeah, this is, and I will show you in a minute, this is the intrinsic chronotype measured at the time of sampling.
Yes,
And
but it could be
the
changed.
question now is that a trait or is that a state? So in other words, how stable is that chronotype? So if I do it in the summer versus the winter, would it be different? Maybe relating to your colleague working on hibernation, and maybe also related to your colleague working on maybe menstrual cycle and contraceptives. Is it stable during the 28 during the monthly phase of women? So we don't know this, but this is all under investigation. And how much depends. So we know that this chronotype or this phase of entrainment can be modulated, because we know if we fly to other time zones, our clock can re-entrain to a new light-dark cycle. So if we come in a new light-dark environment, or maybe I should say more broadly in a more Zeitgeber environment, because it could also be influenced by meal times or so, then we can modulate the phase of entrainment. And the question is probably not whether it can be modulated, more how much can it be modulated, and how stable is it? So can we quantify stability? So, and this is, I just want to show you now the age dependence with this data, and you can see that younger people, as probably many young people can relate, they don't want to come to the lab too early because they have a later chronotype, whereas the PIs who are already older, they cannot understand unless they are in a chronobiology lab where the PI, of course, can understand that for some people it's more difficult to come earlier for the younger people, and as we get older. And look at the end. This is something which we did not expect, and this comes with a note of caution, because you can see the sample size is not so high here compared to the other people. But in questionnaires, it got earlier and earlier. And here we see that when people kind of are over 60, 65, they become later. And we wondered, first of all, whether this is true based on the lower sample size, and if true, what may be the reason. And this goes in the direction of plasticity versus robustness. And one of the hypotheses we investigated using the data was maybe this has to do with work, and people stop working and not have maybe because if you stop working, you're not required to be at work early, and you may not use your alarm clock, and therefore get less light in the early morning hours, which advances your clock. So what we ask is, are you working or are you not working? And this is all. And then you can see that the non-working people have a later chronotype than the working people. And this is all, these models are all adjusted for sex and age again, right? So it's not, this factor is not contributing to this. So we have later chronotype for non-working people. And then interestingly, even if we ask, when did you sample? Was it a day in the middle of the working week, or was it a Sunday? Then we saw a Sunday samplers have 20 minutes later. So this is all cross-sectional, the average people, meaning that even and this is consistent with one of the studies with Martha Merrow did with Dilmo, where she asked in a longitudinal setting, the same person to sample Dilmo on a Thursday and on a Sunday, and depending on the chronotype, when the people have a largely different sleep schedule between Thursdays and Sundays, then the chronotype or the phase of entrainment also was later on the Sunday, even within the same week. And the last data slide I would like to show you is this, where we asked the number of working days and looked at the chronotype, and then there's a clear relation between the more you work 7 days a week, so the earlier your chronotype is. And okay, so there's some people we have only, so as you can see here, most of the data come from a 5-day working week, 2,558, but there are also many people, 435, who do not work, and there are only a few people who work 7 days a week, and there's only a few people. So that means, okay, here, yeah, that we think maybe the chronotype is more plastic than we thought before. That it's depending on the weekday, and so we need more longitudinal studies, and maybe you, with your continuous variable data, can look into that. At least in terms of behavior, you probably see that because this is what is the basis of social jet lag, that people behave differently on a Sunday than on a work day. And what we don't know yet is whether this behavior is also reflected in the molecular clock being more plastic than we thought, or whether this the different behavior. And then the question is the delta between the molecular clock and the behavior something which is correlated with health, mental health, or whatever? Or are people who are more plastic, more resilient towards health-related things? Or so these are completely new avenues to look at. Just one fun experiment, which is I need to state that as the first thing. This is N of 1. Maybe you have seen that. This is me traveling from Berlin to Florida at the SRBR conference in 2022, I think, where we were in Florida. And then I took a hair root sample every day during this travel. And you see here, this is Berlin, and my Dilmo was, you see, it was at 7 p.m., which means in the summer, that was, I was relatively early, so that would be a 10 p.m. sleeping time. And this was this gray was my time in the plane, and I took a sample in the plane, and this is the first day in Florida where it did not change at all. And then at the second day, my phase of entrainment jumped by 4 hours, and then I gradually reached almost the 6-hour time difference between Berlin and Florida. Unfortunately, I had to go back at that time. And then you see that the re-entrainment to Berlin time took much longer, and then that. So that's something, an opportunity for that hair root test, which we could never do with a blood sampling, because there's this would have. So you saw that I did 2 samples in the plane, and because you can put the hair roots in a RNA later stabilizing solution and keep it at room temperature at least for a week.
Okay.
And then and this allows a field study. So I need to stop here and give a summary too. So a hair root cell-based chronotype assay is useful for field studies. Chronotype objectively is normally distributed. Chronotype objective is age and sex dependent, and chronotype is plastic and depends on behavior. And I thank you for your attention. And these are the people from my lab. Here are particularly Christian Gabriel, who was instrumental for the cell project, and Celine and Bert, and these people are for the hair root assays. And as I collaborate a lot with Hans-Peter Herzl's lab, and now we have Luisa Pilz at Charité, who has been with Till Rönneberg earlier and is now a professor for circadian medicine at Charité. And Dieter Kunz is my sleep collaborator. Okay, I thank you for your attention. I'm looking forward to more questions. Thank you.
Thank you, Achim. So, okay, everyone, yeah, please feel free to ask questions. Yeah.
Can you hear me?
Yes.
Yes.
Okay, good. So thank you, Professor Achim, for your wonderful talk. I have questions regarding mostly the biomarkers genes that you identified. So maybe I've missed it, but what genes have you identified as biomarkers, and what are their roles in the cell biology? Are they like linked to signaling pathways, for example?
I'm not sure whether I completely got your question. So you're asking what are the roles of the biomarkers in the. So many of the biomarkers, many of the biomarkers are, as you can see here, many of them here are are clock proteins. Some of them are proteins or genes which are driven by glucocorticoids, because that make them rhythmic. So and others I didn't even look what they're doing, because they are just biomarkers, and I don't have a role for every of them. But I think it's something we should look at. But to serve as a biomarker, it doesn't really matter what they're doing. But I appreciate the question.
Okay, I see. And can you also explain a little bit more what are the difference between these 3 models?
This is something for mathematicians. So the Zeitzeige model